Hey guys, welcome back to my channel. This is part 3 video on red blood cell. Do check out my last two video. So let's get started with the properties of red blood cell. The first property is Rouleaux formation. In the Rouleaux formation, due to the discoid shape of RBC, red blood cells are arranged in a rows, or you can say stalk of coin fashion like this. Red blood cells are piled up on one another, forming a Rouleaux. So this property is called as Rouleaux formation. So here you can see Rouleaux formation of red blood cell. It can occur naturally after blood is collected and allowed to sit for a long period of time. Morphologically can be seen in thick area of blood smear. Pathologically, Rouleaux is only reported when seen in thin area of peripheral blood smear. Associated disease or clinical state. It can be associated with any condition that increases plasma protein, particularly fibrinogen and globulin. Increased plasma protein will coat the red blood cell and cause them to become sticky and result in Rouleaux formation. It can be seen in acute or chronic inflammatory diseases and in multiple myeloma in case of infection. Rouleaux formation can be reversed with addition of saline. On a slide, if you have blood sample and you can see Rouleaux formation, then after adding few drops of saline, then you will see that Rouleaux formation get reversed and you will be able to see independent red blood cells. Rouleaux formation also increase ESR, erythrocyte sedimentation rate. Now our second property is specific gravity. I've discussed this property in last video. You can check that out. Here I'm just mentioning the value. It is 1.092 to 1.101. So the value is greater than 1. So do comment and let me know, red blood cell will sink in water or will float. Third property is packed cell volume. Packed cell volume is the portion of blood occupied by the red cell expressed in percentage. So it is also known as hematocrit value. So you know blood is made up of plasma and red blood cell. So the amount of red blood cell present in the whole blood is called as packed cell volume. So that is around 55%. Fourth property is suspension stability. During circulation, red blood cell remains suspended uniformly in the blood. And this property of red blood cell is known as suspension stability. Next point is lifespan of red blood cell. So the average lifespan of red blood cell is about 120 days. After 120 days, the old RBCs are destroyed in spleen. Spleen is also called as graveyard of RBC. Now, how can we determine lifespan of red blood cell? We can tag red blood cell with radioactive substances like radioactive iron or radioactive chromium, and lifespan of red blood cell will be determined by studying the rate of loss of radioactive red blood cell from the circulation. Moving on to the next topic, that is fate of red blood cell. Old RBCs are destroyed mainly in the capillaries of red pulp of spleen because the diameter of splenic capillaries is very small. Destroyed red blood cells Destroyed red blood cells are fragmented and hemoglobin is released from the fragmented part. Hemoglobin is immediately phagocytosed by macrophages of the body, mainly macrophages present in the liver, spleen, and bone marrow. Hemoglobin is degraded into iron, globin, and porphyrin. Globin is stored and reused, whereas iron combines with a protein called apoferritin and forms ferritin, which is stored in the body and is used later. Whereas porphyrin is degraded into bilirubin, which is excreted by the liver through bile. So this was all about fate of red blood cell. Daily 10% of old red blood cells are destroyed in normal healthy adult. Okay. Function of red blood cell. So the first function is transport of oxygen from the lung to the tissue. Hemoglobin in the red blood cell combines with the oxygen and forms oxyhemoglobin, and around 97% of oxygen is transported in the blood in the form of oxyhemoglobin. Second function is transport of carbon dioxide from the tissue to the lung in the form of carb hemoglobin, where hemoglobin combines with carbon dioxide to form carb hemoglobin, and about 30 to 40% of carbon dioxide is transported in this form. Rest 60% is transported in the form of bicarbonate from the tissue to the lung. The third function is buffer action in the blood. Hemoglobin function as a good buffer by regulating hydrogen ion concentration and thus maintain acid base balance. Fourth function is in blood group determination. RBC carries blood group antigen like A antigen, B antigen, and Rh factor. This helps in determination of blood group and enables to prevent reaction due to incompatible blood transfusion. So this was all about function of red blood cell. Thank you for watching. Stay tuned for the next video.